Size matters. New data reveals cell size sparks genome awakening in embryos. Transitions are a hallmark of life. When dormant plants flower in the spring or when a young adult strikes out on their own, there is a shift in control. Similarly, there is a transition during early development when an embryo undergoes biochemical changes, switching from being controlled by maternal molecules to being governed by its own genome. For the first time, a team from the Perelman School of Medicine at the University of Pennsylvania found in an embryo that activation of its genome does not happen all at once. Instead, it follows a specific pattern controlled primarily by the various sizes of its cells. The researchers published their results this week as the cover story in Developmental Cell. In an early embryo undergoing cell division, maternally loaded RNA and proteins regulate the cell cycle. The genomes of the zygote, a term for the fertilized egg, are initially in sleep mode. However, at a point in the early life of the embryo, these zygotic nuclei wake up, and expression from their genomes takes biochemical control over subsequent embryo development. But how an embryo recognizes when to undergo this transition has remained unknown. How an embryo hands over control of development from mother to zygote is a fundamental question in developmental biology, said senior author Matthew C. Good, Ph.D., an assistant professor of cell and developmental biology and of bioengineering. Previously, it was not appreciated that different regions of a vertebrate embryo can undergo genome activation at different times, or how directly cell size regulates the awakening of a zygote's genome. Different hypotheses have been offered over the last 40 years to explain how an embryo discerns when to turn on the new genome of individual cells within the zygote, but it was the Penn team who nailed the mechanism and answered this key question. Using single-cell imaging of embryos from the African clawed frog Xenopus laevis, they found that cell size was the key parameter governing the start of genome activation in zygotes. Cells must achieve a threshold size to initiate large-scale transcription of their own proteins. By generating miniature embryos, the team demonstrated that changes in cell size control the timing of genome activation. The results of this study have a number of important implications for the basic understanding of how an embryo develops in its earliest days and for the field of developmental biology in general. The Penn team believes this finding could impact how other investigators approach their own research on genome activation and screening for maternal factors that are necessary to control the fidelity of early embryo development. To gain new insights, zygotic transcription should be measured at a single-cell level, said first author Wei Chen, Ph.D., a postdoctoral fellow in Good's lab. This approach helped us to not overlook the influence of the spatial organization of an embryo's cells on the maternal zygotic transition. The decision to initiate the zygote's genome is made at the level of individual cells, not the entire embryo, which has changed the Penn team's view of the developmental process. Evolution has co-opted cell size as a regulatory mechanism to control a critical transition in embryonic development, a paradigm that may extend to other areas of biology in which cell size varies, said Good. He and Chen plan to continue this work by measuring genome activation in zebrafish and mice to see if this new perspective holds true in other species. This work was supported in part by the Burroughs Wellcome Fund, the Charles E. Kaufman Foundation, the March of Dimes, and the National Institution of General Medical Sciences. Penn co-authors on this paper include Sean C. Little, Ph.D., an assistant professor of cell and developmental biology. Dinosaur bones are home to microscopic life. Bad news, Jurassic Park fans, the odds of scientists cloning a dinosaur from ancient DNA are pretty much zero. That's because DNA breaks down over time and isn't stable enough to stay intact for millions of years. And while proteins, the molecules in all living things that give our bodies structure and help them operate, are more stable, even they might not be able to survive over tens or hundreds of millions of years. In a new paper published in eLife, scientists went looking for preserved collagen, the protein in bone and skin, in dinosaur fossils. They didn't find the protein, but they did find huge colonies of modern bacteria living inside the dinosaur bones. This is breaking new ground. This is the first time we've discovered this unique microbial community in these fossil bones while they're buried underground, says lead author Evan Seda, a postdoctoral researcher at the Field Museum. And I would say that it's another nail in the coffin in the idea of dinosaur proteins getting preserved intact. Seda began researching organic molecules in fossils as part of his doctoral thesis at the University of Bristol. My PhD work focused on how soft tissues fossilize and how these materials break down. Some molecules can survive in the fossil record, but I suspect proteins can't. They're unstable on those timescales in the conditions of fossilization, explains Seda. However, some paleontologists have reported finding dinosaur bones that contain exceptionally preserved traces of the protein collagen, along with soft tissues like blood and bone cells. There's been an uptick in interest in these supposed dinosaur proteins, says Seda. So, he set out to try to independently verify the presence of collagen in dinosaur fossils. Seda took pains to collect dinosaur fossils under as sterile conditions as possible so that new proteins or bacteria wouldn't be introduced to the fossils and skew the results. He took a pickaxe, saw, blowtorch, ethanol, and bleach out to Dinosaur Provincial Park in Alberta, Canada. There's a single layer where there's practically more bone than rock. It's ridiculous how concentrated the bones are, says Seda. A site with lots of bone was key, because a slow, meandering dig would open up the fossils to more chances to be contaminated by the surface world. To collect these bones in a very controlled, sterile way, you need a dig site with a ton of bone because you have to find the bone quickly, expose just enough of one end to know what it is, then aseptically collect the unexposed bit of the bone and surrounding rock all in one. Seda collected 75-million-year-old fossils from Centrosaurus, a smaller cousin of Triceratops, and then took the bones back to various laboratories to examine their organic composition. Seda and his colleagues compared the biochemical makeup of the Centrosaurus fossils with modern chicken bones, sediment from the fossil site in Alberta, and thousands of years old shark teeth that washed up on the shore of Seda's hometown of Ponte Vedra Beach, Florida. We visited multiple labs, and the different techniques gave us consistent and easily interpretable results, suggesting that the aseptic collection was sufficient, says Seda. They found that the Centrosaurus fossils didn't seem to contain the collagen proteins present in fresh bones or the much younger shark teeth. But they did find something else. We see lots of evidence of recent microbes, explains Seda. There's clearly something organic in these bones. And since the lab work indicates that Seda's anti-contamination measures worked, these organic materials must have gotten there naturally. We found non-radiocarbon dead organic carbon, recent amino acids, and DNA in the bone. That's indicative that the bone is hosting a modern microbial community and providing refuge, Seda says. He thinks, as others have previously suggested, that the modern microbes and their secretions, called biofilm, are likely what other researchers have seen in fossils and reported as dinosaur soft tissues. I suspect that if we began to do this kind of analysis with other specimens, it would begin to explain some of the so-called dinosaur soft tissue discoveries, he says. Surprisingly, the modern microbes present in the dinosaur bones aren't quite the same run-of-the-mill bacteria living in the surrounding rock. It's a very unusual community, says Seda. 30% of the sequences are related to Eusebia, which is only reported from places like Etruscan tombs in the skin of sea cucumbers, as far as I know. Seda and his colleagues aren't sure why these particular microbes are living in the dinosaur bones, but he's not shocked that bacteria are drawn to the fossils. Fossil bones contain phosphorus and iron, and microbes need those as nutrients. And the bones are porous, they wick up moisture. If you were a bacterium living in the ground, you'd probably want to live in a dinosaur bone, he says. These bacteria are clearly having a jolly good time in these bones. The discovery could help further the emerging field of molecular paleontology, says Seda. It's one of the new frontiers of modern paleontology. We are beginning to undertake a very different kind of fossil hunting. We're not just looking for bones and teeth, hoping to find new species, we're doing molecular fossil hunting. It opens up an entirely new line of evidence by which to study life in the past. Molecular fossils can tell us things we never thought we'd be able to investigate. Distinguishing what is modern from what is ancient is important. This study was contributed to by authors from the Field Museum, Princeton University, the Chinese Academy of Sciences, the Royal Tyrrell Museum, the University of Bath, the Foundation for Scientific Advancement, Revive and Restore, Johns Hopkins University, the American Museum of Natural History, Newcastle University, the University of York, and the University of Bristol.